Greetings, faculty, and welcome to another WIS Learning and Technology conversation. Last time we spoke with Tom Heilman on his use of the iPad in marking papers without papers. And this week I'm going to move into the science department, and I have here in the studio with me a biology teacher, Paul Chatelain, and he's going to be talking about the microscope and how it has evolved in his classroom in sort of almost in some ways a social network, in that he's now been able to capture some very interesting organisms and display them and show them on a YouTube channel that is now gaining popularity with a number of views, and he has some very cool ideas about how this could be implemented in other schools and shared with other educational institutions. So without further ado, I want to introduce here Paul. And Paul, would you mind giving us a brief introduction to yourself and perhaps, I don't know, your educational philosophy towards the sciences and more specifically biology?
Sure, yeah. Well, I've been here at WIS now for 3 years, and I think that the sciences here are probably one of our stronger programs, as is probably evident in the fact that we have a lot of kids who in the diploma program end up taking a lot of the higher level classes. They do really well in them overall. In terms of my educational philosophy, I think that science is not more important than any of the other subjects, but I think it's one of the crucial aspects because it teaches kids not just how to think in unique ways, but also how to kind of shape their worldview around more than just social aspects, and they get to think more in terms of how things are working together and put together in non-social ways. Not to say that the social aspect shouldn't be a part of the sciences either.
Great, and it's no secret to anyone that the microscope is perhaps maybe the landmark or the essential tool in any kind of beginning biology class to really understand cell division. And I'm no scientist, but I remember using microscopes in my biology classes. And could you talk a little bit about how your approach to getting the kids accustomed to using the microscopes properly and being able to identify what they're looking at?
Yeah, well, it's actually a process that starts well before they get to me. They start in 6th and 7th grade with Kusum and Trus, and by the time they get to me, they've had a little bit of experience with it. But what I do is I just kind of refresh their knowledge in terms of the parts and what they are and how they work and how the microscope works. In physics, they actually go over some of the optics involved in it. So in biology, we really get to just hone in on looking at things under the microscope. So what I try and do is I try to show kids how life is everywhere. You can find it in a teaspoon of dirt. You can find it in a cup of water out from the back. And so what we do is we go around campus and we find places where the kids before this don't necessarily know where life is. We take samples and then we bring those samples back into the class and we try to find things under the microscope. So we start there and then we work towards the identification, which is a challenge for the younger kids for sure.
Now, in the past, the microscope, I think, has kind of been it's been a real sort of intimate experience between the person looking in on the organisms and that one individual. But now, you've got a nice little toy here that allows you to almost socialize the microscope in a sense that everyone in the class can simultaneously see what is going on under there. And could you talk a little bit about, let's talk about the gear. Like, what is this toy you have? It's not really a toy, obviously, but this camera microscope, what is this?
So we have, when I first came, we had a very simple, it was essentially just an adapter where you could slip it into the microscope tube in place of the eyepiece, and it didn't have very good resolution. And actually, some of the videos that we're going to talk about here on the channel, the YouTube channel, are from that original microscope. But now what we have is, the brand is Modic, and it's the BA310, and it's got a digital microscope camera that's embedded within it. It's got all kinds of objectives. We can go from about 40 times magnified to over 1,000 times magnified. We can change lighting, and we can play with contrast. All of those features are just embedded right in the microscope, and it's really it's a really nice tool to have. Toy maybe, but tool I like to say.
Absolutely. Now, if I remember correctly, at first you were kind of amassing several gigabytes of video, and then finally you kind of came to this idea of how to share it, and then this idea of developing a YouTube channel and kind of documenting what's under there, and perhaps adding a little more description about the magnification and to make it actually searchable. Could you talk a little bit more about your... In fact, let's just dive right into your YouTube channel here and visit some of these really interesting creatures that are here.
Yeah, sure. So what I did was I had, as you mentioned, just hours and hours of videos that I had taken under the microscope. And here's some examples of some of them. I believe they're chronological here. So here's some of my newer ones, and you can see they're a little bit crisper. And I'll play a few here in a second. And I have, quite a few. But regardless, when I first started, I just had videos. Let's play one of my really early ones. So this is one of the first ones. And I didn't have anything to do with them. I didn't really know what to do with them. I just sort of knew intrinsically that it would be a valuable thing to do. And I think this is a nice example of where the technology really supported what I wanted to do. Instead of the technology coming first, it just grew out of what I needed to do. And what I needed to do was share in some way these videos, and then also be able to use the videos that I had in class. And of course, it's not working right now.
Yeah, a little bit of bandwidth issue perhaps, but...
So anyway, I was taking all these videos. We'll see if we can get maybe another one to work. And when... Yeah, here's a nice one. And this is actually a sample that was taken from the fountain just outside of the dacha. These are just a particular kind of algae called Volvox. So what I did was I just started taking videos and amassing them on this half terabyte hard drive that the library was kind enough to furnish the bio department with. And then it started to grow into, wow, I wish that I could access that video that I took last week and show it to the kids and share it with the kids. And then, as you mentioned, the social aspect of the microscope, it used to be, until fairly recently, very individual. Someone would be looking under the microscope, and that was the only person who could actually see what was going on, who could actually appreciate what was going on. And ultimately, for the kids, really understand what was going on, because I can look through it and know what I'm seeing, and I can try and describe what I'm seeing to the students. But ultimately, it's very difficult for them to be able to transfer some description or even a picture into something that's useful. So here's another one that we took. This is Vorticella. Let's see. These are found pretty heavily in any of the samples that we find. Soil, we find them in the worm bin as well. Apparently also not going to work.
Yeah,
but
looks like we're having some small network issues. But I'm curious about what sorts of questions do let's go back to the Volvox one. We saw that one play earlier, right there. So what kind of questions does this prompt with students? Like when you're actually displaying something like this, how do you contextualize what they're looking at? So let's just hit pause here and kind of...
Well, so Volvox are a type of algae, and when we're doing something like this, what we can do is we can use this as a way to explore what is algae and why is algae important. These are particularly interesting. There's a few things that I'll just point out biologically here. There's actually colonial, so these are each of the little specks, the little dots that my cursor's over here, are individual cells, and then they secrete a fluid-like, jelly-like matrix into which they can hold on to each other. And then each of these smaller, darker green circles here are sub-colonies that will break off and actually spawn new, smaller little Volvox colonies. So you can use these as a way to illustrate how cells can work together to actually protect. If you notice, these are in the inside, so they're protecting the growing colonies, but they're also able to rotate so that cells that have a better exposure to the sunlight will move so that other cells that didn't have quite as much of a chance will have better exposure. And then you can also talk about how reproduction is important here in terms of all living things, and how do you find this? What are the ways that you obtain a sample like this? So there's lots of opportunities.
Way cool. Now, do you have any kind of interesting anecdotes with your students on using this camera and displaying? Like, is there any particular video that stands out or has a story kind of behind it that you'd be willing to share with us.
Yeah, sure. Let's go back to the channel. So there's, I could talk about any of these, but there's a few in here.
We'll cross our fingers that
the one I'm looking for will work. Yeah. So this one is actually very cool. Cool is a good word for it. Oh, come on. Let's hope it works. So, good. All right. So this is a video that I took from the worm bin. Can I make this full screen?
Absolutely.
Okay. So, from the worm bin, the liquid that just leaches out of rotting things gets collected in the bottom of the worm bin. And this is a particular type of worm. And you can actually see...
Can I interrupt you for a second?
Sure.
It seems like you're panning. Does this also allow you to pan the camera up and down? I see camera movement.
Yeah. So you can actually, there's a little track bar almost where I can move the slide around. So this thing can actually be quite active, and I can do a fair job of keeping pace with it and following along an active animal.
Cool. And this is not really cooperating today, but here we go again.
Yeah,
okay.
It should work. All right, so this, again, this is a worm, and the particular type is called Alosoma. Maybe it's stopping at 5 seconds.
Not liking our full screen mode, perhaps.
Yeah, let's try that in. Okay, so here towards the front here, the anterior end would be the mouth, and then the anus at the end, so it's got a complete digestive tract. And then at 36 seconds, so I'll do it just before that, you actually get to see this guy eat a much smaller single-celled organism. And if you look at, there it just did it. So here, I'll back it up again so that you can get a better look at it. But this was an opportunity where, they say that lightning doesn't strike twice. There it goes.
Wow.
So lightning did strike twice here because, believe it or not, I saw 2 of these instances within about a minute of each other. This same worm was under my camera, and it ate, in that case, a single-celled protist. And I said, oh my goodness, and I was actually with the 11th graders at the time, which were not using microscopes. But I said, oh, you guys have got to see this. This is too cool to miss. And so what we did was we put it up on the big screen. I started recording, and that's why there's so much dead time. There's 36 seconds of nothing interesting. Because I said, maybe, just maybe, it'll do it again.
Yeah.
And it did. I got lucky, and the kids thought it was really cool. And this was for a long time up on, I get to post this on my Moodle page as well. So it's got 35 views, which is pretty fun.
Fascinating. So that leads me to my last question: is what's next for the WISP Bio channel, and how do you see kind of this socialization of microscopes being able to help schools kind of in the future? Obviously, this piece of equipment perhaps isn't the most affordable, and that maybe not all schools could buy something like this, but the videos that are out there, and this could be shared prolifically around the world, and I'm just wondering, what do you foresee? What would you like this to become?
No, it's a great question. In terms of, so if we just look right here, when a video finishes playing on YouTube, you're left with all of these links. So here we have something that has absolutely nothing to do with the microscope or education or anything useful. And then we look, and here we see all kinds of other links that are appearing that are in some way related. So ultimately, the goal is to create essentially almost like a field guide for WIS. Here are things that we can find at WIS. Here are videos of them. That way, when a student says, gosh, I just saw this thing under the microscope, then they can go to the WIS YouTube bio channel and they can search, and they can maybe have some descriptors, right? They could say, oh, well, I saw these things and it looks like this and it was this color and it was about this size. And if we could make it a searchable catalog, a nature guide, where the kids can then go and find things. So that would be step one. And it would then become a very valuable resource for the students. Now, on a sort of larger scale, if that could work, then it would be really nice for these other links here, none of which are mine directly, to be more of, the internet, in my opinion, is just a great way to share information. Part of the problem is that there's no way to know how valuable that information is, how correct it is, who's putting it up. So if there would be some way that either Google or YouTube could actually pay somebody to just peruse, and I know peruse is probably not the right word, but scour the internet and find videos that are related to the Alosoma that we showed, or Volvox, or any of the other types of things, and compile them and help give schools, like you said, that don't have the money. I mean, this is downright expensive, the microscope camera that we have. Then they would be in a much better position to be able to just share information, because that's ultimately the goal here. You want to be able to share and inform and allow people to have an experience they might not.
And now your channel here is approaching, I think it's nearly 3,000 views, and I think it's fantastic that a school as relatively small as ours is definitely indicating that it's not just us consuming these videos, that there's several other schools that have come across these and are using them, and I think it's really neat. So
yeah, if we look here just real quick, that's the most popular one. This is the one that's been up longest, which I'm sure is why it's most popular, but I've got almost 1,200 views, which is pretty cool.
Yeah, and it's definitely not how I spend my time watching YouTube, but it's fantastic that
someone is, right?
someone is doing it, and they're learning biology on YouTube, which is fantastic. Well, Paul, thanks so much for joining me on the program today, and perhaps we'll have another chance to talk about another science-related topic. So,
thank you, Richard.